is to get it on that screen, you have to move it in that direction. To get it on that screen, you have to move it in that direction. And it probably messes with your head. You don't know where it's going. Anyway, I think we're good. Right, so I'm hoping that nothing goes wrong with this before Michaela joins us to talk about the MHC shortly. But before that, innate immunity. Does anybody have any questions about things that were in the innate immunity lecture? There was a question about the inflammasome, which we can try to, I can try to find, let me see. Oh, this is big as well. I need to do this on another computer. That. Oh. Ah, here we go. I'm gonna have to just scroll through these, get to the slide I want slowly. There we go. So somebody had asked about a bit of an explanation about the inflammasomes. Let me stand where I can see the microphone. So basically, the inflammasomes are an important part of the immune response, particularly for the generation of these two cytokines, so IL-1 beta and IL-18. And the reason, I think, the reason why it's so complicated for these two cytokines isn't really fully understood. It's probably a mechanism to make sure that inflammation is controlled, and we only get inflammation when inflammation is needed, because as we discussed previously, inflammation per se is great if you have an injury or if there is an infection, great. If there isn't, you probably don't want a great amount of cytokines, so on, going around your body. So these two cytokines are essentially synthesized and secreted in two different stages. So they're first of all synthesized as a pro-molecule, which is completely inactive, needs to be cleaved to make the active molecules of IL-1 beta and IL-18. Now, the way that this works is it's a two-stage process. You first get the synthesis, and the synthesis of these molecules is much the same as any other synthesis of co-stimulatory molecules, regulation of MHC, other pro-inflammatory cytokines, following the recognition of PAMPs and pathogens by a pattern recognition receptor. We get the signaling pathway via MyD88, and we get the activation of NF-kappa B, the nucleus. That activation of NF-kappa B leads to the synthesis of the molecules, the pro-molecules of IL-1 and IL-18. But then you need a second signal, and the second signal is provided by the inflammasome. So this recognition of a pathogen by a pattern recognition receptor is not enough to get these molecules synthesized and cleaved to make an active form. The second signal is provided by any number of other things, such as uric acid crystals, such as increase in ATP, alarmins from your own cells, cellular damage, can lead to this signal 2, which leads to lysosomal damage, and that signals the cell to synthesize and assemble the inflammasome. Yeah.
Increase in the concentrations of uric crystals associated with like inflammation and infection.
Yeah. So I think the most common condition that leads to the accumulation of uric acid is gout. Have you heard of gout? Does everybody know what gout is? So, gout's quite an interesting disease because it's always associated with fine living and drinking good wine and eating rich food, and and men get it more than women. But actually, most people who suffer from gout have got a enzyme deficiency, which doesn't allow them to break down their crystals like most people do. And what is quite interesting is that women are hormonally protected from gout until they get to the menopause, and then all bets are off, and they are just as likely to suffer from gout as men do. So you're absolutely right. And one of the hallmarks of gout is extreme inflammation, massive swelling of usually the feet, although you can get it in hands and in elbows, massive swelling of the feet. So yeah, massive inflammation. So that second signal gets this inflammasome assembled. You need your NLRP3 molecule, you need your adaptor protein, and you need the pro-caspase molecule, because caspases also need to be cleaved. They don't just exist. And that leads to the synthesis, the cleavage of pro-caspase 1 into caspase 1, which is the enzyme that then cleaves your pro-IL-1 beta into IL-1 beta. So this alone will not lead to active IL-1 or IL-18. And as I said, it's probably a control mechanism to make sure that we don't actually make IL-1 and IL-18, which are very potent inflammatory molecules, when we don't need to do so. So it's kind of a bit of a belt and braces approach to it. So I hope that makes that a little bit clearer. Does anybody have any other questions about the innate immune response that we went through? No? Excellent, that's good. That was all super clear then. So I have a question for you, I guess. So we talked a little bit at the end about how the innate immune response will affect the adaptive immune response. Does anybody give me an example of how that might happen? We've talked, you kind of talked a little bit about it today already. What do we need to happen for T cells, for example?
Because the innate immunity is what produces the co-stimulatory molecules that are going to have the effect on the T cells and the B cells, which is the adaptive.
Exactly right. So you know that if you only have one signal for T cells, for T cell activation, they don't do anything. So we need co-stimulation. Co-stimulation is really important. One of the ways that we get upregulation of co-stimulatory molecules is via the recognition of PAMPs, or pathogens by pattern recognition receptors. So that's one way that we do that. Any other way that anybody thinks that they might be able to, antigen presentation? Yeah. So how do you think the innate, what role does the innate immune system have in antigen presentation?
Migration to the lymph nodes to present.
Yeah, exactly.
Didn't hear what you said.
Antigen presentation was the thing, and we have got migration of cells, and we also the cells themselves. So the dendritic cell is classed as part of the immune system, and the dendritic cell is the cell that needs to present antigen to the certainly to the naive T cell in order to start the whole process off. Of course, what else do dendritic cells do? They don't just present antigen, they might make something. Might they make? What tells cells what to do and what to be? Cytokines. Okay, so we might activate our T cell. We might also need cytokines to be produced to tell our T cells what they're going to be when they actually fully mature. So there are lots of ways in which the innate immune system will help or drive the adaptive immune system, and that's kind of quite logical, because the innate immune system happens first. Does anybody think of a way that the adaptive immune system might influence the innate immune system?
cytokines as well.
Exactly. Exactly right. So your CD4 positive T cell, Th1 cell, will make. Anybody know what the signature cytokine for Th1 is? Well, you haven't done that yet, so I'll let you. Interferon gamma. And interferon gamma is a very potent activator of macrophages. So a macrophage will do killing, because that's what it does. But if it's activated by interferon gamma, it will do killing even better. Same with antigen processing and presentation. Interferon gamma upregulates the molecules involved in that. So that's an example where the adaptive immune response feeds back and potentiates the innate immune response. Yeah? So that's kind of how the two things fit together. So although in our heads, it's very easy to think, like, this happens, then this happens, and this happens, and then we're done. We're not actually done, because the whole thing starts to feed back from the beginning until, of course, the most important part of the immune response happens, and that is turning it off. Because if we didn't have a way to turn off the immune response, the first cold you ever got, that would be done for. You'd just be inflamed for the rest of your life. And that's obviously not the case. You end up getting over your cold, and your immune response goes back to normal. So, okay, I see. Michaela, can you hear me?
Yes, I can.
Excellent. I'm going to try and unshare my slide somehow. This is quite a thing. Yes. So, Michaela, thank you very much for joining us. You can hear us?
Yes, I can.
Can you see us?
Yes, I can.
Excellent. So, Michaela, there we have a whole group of people assembled here with questions for you about your lecture. I have some that have been posted on our Padlet that we're using. So let me just get to those. Okay. So I think we have a question about MHC class 1 and class 2 complexes, and how are MHC class 1 molecules more specific than MHC class 2 complexes, and how does this affect MHC peptide binding affinity?
Okay. Let me see if I have a slide that I can show you the the classic allocation here. And can you see me?
We can see you, yeah.
Good. Then I share with you.
It's going to come. Oh, there. Oh, marvelous. And we can see your slide as well. Very good.
Good. So here is a representation of what is the structure of the MHC class 1. And can you see my mouse as well?
Yeah. Yes, we can, yeah.
So this in the panel A is the MHC class 1 bound to a reference peptide, and the panel B is the MHC class 2 bound to the peptides. In this case, the peptides are in this gray color. And you can see in D and E how the peptide is allocated usually within the cleft of the MHC class 1, as well as on the contrary is the peptide for the cleft of the MHC class 2. So as you can see, one characteristic is that the peptide is really included in the cleft of the MHC class 1, that then this leaves less freedom of the peptide in terms of affinity between the cleft of the MHC class 1, so that specific allele, and the sequence of the peptide. On the contrary, for the MHC class 2, the position of the peptide is more loose, and can even, so is not limited within the cleft, and this leaves more freedom or flexibility in terms of sequence that can be bound to the MHC class 2 compared to the MHC class 1. One outcome of this different characteristic and therefore different binding affinity is that the binding affinity between the peptide MHC class 1 can be predicted with more precision than what is so far possible for the peptide in the MHC class 2 molecules. This also means, as you can see here, is also represented how the TCR alpha beta are recognizing the peptide in the MHC class 1 and the MHC class 2. So also that implies that for the TCR alpha beta, they recognize the peptide bound to the MHC class 1. It is mainly the CDR3 region that define the specificity of the TCR for the peptide MHC class 1 molecule.
Okay, thank you. We have another question, which is a little bit about MHC in pre-transplantation testing. So you may or may not know the answer to this, which MHC molecules are the most important for looking at pre-transplantation testing, and how do we identify MHC epitopes? How do we know which epitopes MHC bind to?
I'm sorry, I didn't understand the question.
Okay, let's take the second question, part of the question first. Probably the easiest. How do people go about identifying what peptide epitopes, or what epitopes are important for MHC binding? How do we know what the epitopes are?
So I also remove 1 second. I stop to share. So first I would like define the term epitope. So as I was saying, so the peptides that are presented, for example, by the MHC class 1 complex, they are defined as antigenic peptides because they arrive from an antigen and are presented at the cell surface by the MHC class 1 molecule. The simple presentation, so only the presentation of the peptide by the MHC class 1 molecule is not sufficient to define a peptide as epitope, because then an epitope is a peptide that is presented that is able to trigger a CD8, so is recognized as the TCR of the, for example, CD8 T cells, and is able to trigger an activation of the T cells. Therefore, how we can identify the epitopes that are presented by MHC class 1? So the epitope per se means that there is an so there is a CD8 T cell activation, so that the epitopes, they are immunogenic, so they are able to trigger a CD8 T cell response. This can be tested, for example, by performing experiment where we have the peripheral blood mononuclear cell of donors, for example, let's say cancer patients, that are co-cultured with professional antigen-presenting cells that express the specific epitope, or that they are pulsed with the synthetic peptides. So we can add the synthetic peptide to the cell culture where we have the antigen-presenting cell with a specific MHC class 1 complex variant, and we have the CD8 T cells in the peripheral blood mononuclear cell of, for example, cancer patients. And then with different type of assay, one of the most used is ELISA or ELISpot, that measured interferon gamma release by the CD8 T cells when they recognize, if they recognize the peptides. If this is the case, then we are in a situation where the peptide is most likely, well, I mean, it's most likely, when the peptide could be an epitope. Okay, but again, in order to be an epitope, the peptide should be able to trigger a response, ideally in vivo. Then, this is to define an epitope. We can also identify which peptides are presented by the MHC class 1 molecule, and this could be done in different ways. One of the most used method at the moment is through the analysis via mass spectrometry of the HLA or MHC class 1 immunopeptidomes. So the peptides that are bound to the MHC class 1 molecule. So we take cells, we lyse it, we pull down the MHC class 1 molecule in condition that keep the peptide that is bound to the MHC class 1 molecule in the cleft. And once that we pull down the MHC class 1 with the peptide, then we use condition that they strip the peptide from the MHC class 1, and the peptides are then analyzed measured by mass spectrometry, and they are analyzed with standard proteomic approaches. And this gives us an overview of the MHC class 1 immunopeptidome of a given cell or of cancer, for example, or cells that have been infected. With the current methodology for proteomics and the most accurate mass spectrometer that are available, from a cell line or a cancer, we can identify between 4,000 and 40,000 peptides. The fact that they are presented by the MHC class 1 molecule, as I was mentioned before, it doesn't mean that they are recognized by the TCR. For example, you should always consider that there is a mechanism of central tolerance. Therefore, many TCRs that recognize self-peptides are then not present in periphery because it's been eliminated during the negative selection in the thymus. So the fact that a peptide is potentially an epitope, it doesn't mean that it's really an epitope, because if there isn't a TCR, then the peptide per se is not an epitope.
Thank you. Any other questions for Michele? I think, Michele, there's still a little bit of confusion about cross-presentation. I don't know whether you have any thoughts on that. We've had Kitano on earlier giving us his thoughts about cross-presentation. I don't know whether you give any insights into.
Yes. Yeah, even in other modules, so the cross-presentation is always complex, and the reason is that it's a different mechanism. It's a difficult mechanism to analyze, because dendritic cells are not easy to grow, so it's always tricky, and there are alternative pathways that also trigger in the last 20 or 30 years quite intense discussion about how relevant they are. So one of the important things that you should remember is that, yeah, the cross-presentation is mainly mediated by dendritic cells. The dendritic cells, the cross-presentation then lead to the presentation of exogenous antigen through the MHC class 1 antigen presentation pathway. That implies that they are most produced by, so the peptides that are bound to the MHC class 1 are most produced by proteasome. Okay, one characteristic of the dendritic cell is that the proteasome isoform that are present in the dendritic cells, they are mainly immunoproteasome. Okay, and so the theory is that in order to have a proper response of a CD8 T cells, so priming by these dendritic cells, then ideally you have the same peptide that is presented through cross-presentation is then also the peptide that is recognized in the target cells. So there is still kind of unclear how then for many peptides that are not well presented by immunoproteasome, then we can have a good priming and of the CD8 T cells, and then the recognition of the peptide in the target cells.
Okay, thank you. Any other questions for Ms. Taylor? Online?
I've got a question.
Okay, yeah, go ahead.
So, for the protease catalyzed peptide splicing part, you talked about cis and trans, but I was a bit confused. So is this when the peptide splicing is on the same antigen, or is it when the fragments are combined from two different antigens? Because I didn't understand that.
Yeah, so the cis-spliced peptides are those that they derive from fragments that are present in the same antigen. And the trans-spliced peptides are those that derive from fragments that they derive from two different antigens. So for the MHC class 1 antigen presentation pathway, that is mainly the peptides are mainly produced by proteasome, that is, according to our results and other groups, they are quite efficient in catalyzing peptide splicing reaction. So far, the majority of the spliced epitope that has been described are cis-spliced epitopes. The relevance of the trans-spliced peptide in the MHC class 1 pathway is still quite a controversy, so I would suggest that maybe we don't even discuss that. But the point is that for the MHC class 2 pathway, where the peptides are not produced by proteasome, but are produced by other proteases, there's been, since 2016, many examples of what they are called insulin as a hybrid insulin peptide, that they are epitopes presented by MHC class 2 molecules of the pancreatic beta cells. They are, so the theory is that they are so they trigger an autoimmune response in type 1 diabetes. And the insulin, hybrid insulin peptide, they are almost all of them trans-spliced peptide, because they are the fusion of one portion of insulin or proinsulin and another antigen that is characteristic of the beta cell in the pancreas. And there's been many examples, both in mouse and human type 1 diabetes patients, where these trans-spliced peptides, they trigger a specific response. So it depends so in terms of the difference between cis and trans is, as I described, but then the relevance in one pathway and the other is different. So trans-spliced peptide, at the moment, they are better known and most studied in the MHC class 2 pathway, and the cis-spliced peptide in the MHC class 1 pathway.
Okay, does that answer your question?
Yeah.
Excellent. Any other questions? Michaela. Michaela, you've stunned them into silence with your MHC lecture. They all know it all. They're all clear. Everybody's clear. That's good. Sure, this is your last chance. I think it's Friday afternoon and everybody's tired. Okay. Anybody online want to ask Michaela a question? You've all been very quiet online. I know you haven't been able to hear us for most of the afternoon, but now you can hear us. Does anybody have a question? No? Any more in the room going once, twice? No, Michaela, I think you are free to go, having answered the questions. Thank you very much for giving us time. Bye.
Nice weekend. Bye-bye.
Have a nice weekend. Bye. So, guys, just before we go, there was something I have to say. What fell out of my head? Yeah, so from now on, we will be doing these online, but there will be some mornings when you have a Friday morning session, a research skills session in person. Now, some of these won't finish until 1 o'clock. Might make it tricky for you guys to get to wherever you live in order to join the online tutorial. But because we had planned to do these as high flex, we will always have a room booked. So we will let you know which room, or you will know which room that is. And so you will be very welcome to come to that room and join the online session in that room, because that room will be only designated for the MSc. You might need to bring headphones, otherwise it might be a bit weird with all of the sounds coming out of your individual computers. But so if there are times when people can't get to somewhere else to do the online tutorial, then that this room will, or it might not be this room, but a room will be available for you to go to. Yeah. The second thing I had to say was, I don't know, I couldn't remember whether Dina said it because I was trying to do the AV, but personal tutorials. Hopefully you all met your tutor. I think some people didn't, and it would be good if, and this is for those online as well, because we're not entirely sure who's supposed to be here and who isn't at the moment. But if you're not coming to the tutorial, if you would be kind enough to let your tutor know so that they're not hanging around waiting for someone who doesn't show up, that'd be great. And I know we've got another tutorial next week, so, or the week after, I can't remember quite when. So if you're not going to be able to make that, then please let your tutor know. It'd be great. So with that, thank you very much for your patience with this that we have here. It kind of worked when we had the two people online, so that was good. It took a bit of a messing about, but now we've decided we're going fully online, so we don't need to do this anymore. I wish you all a lovely weekend. I think it's supposed to be reasonably decent weather, so maybe be able to get outside, not stay inside and work. All right, have a good weekend. Thank you. I'm going to try and do something with this. Thank you to the guys online. Bye.
Bye.
Bye.